It's exactly 11, so we'll go ahead and get started. It's my privilege today to introduce our seminar speaker, Dr. Richard Kuhn. Dr. Kuhn comes from us from West Lafayette, Indiana, where he serves as the director of the Purdue Institute of Inflammation, Immunology, and Infectious Disease, and he's a leading expert in the virology community. As you'll see today, his research team employs structural and functional approaches to investigate various aspects of the virus replication cycle. Dr. Kuhn received his PhD at State University of New York at Stony Brook and did postdoctoral training at Caltech. And he came to Purdue in 1991 as an assistant professor in the Department of Biological Sciences and has been there ever since, working his way up to chair of the department and now institute director. Dr. Kuhn has published over 180 papers, many of which are in high-impact journals such as Science and Nature, and he serves as an associate editor on the editorial board of leading virology journals. He's also served on several NIH study sections as an expert peer reviewer. Dr. Kuhn is most well known for his work on two closely related families of viruses. These are the alpha viruses and the flaviviruses, and most people don't know these viral families by name, but you're certainly aware with the type species, especially the human pathogens, chikungunya virus, dengue virus, yellow fever virus, West Nile virus, and Zika virus. Dr. Kuhn has spent many years elucidating the mechanisms by which these viruses replicate their nucleic acid genomes and assemble virion particles, and these are topics that are very near and dear to my heart because it's what my lab also works on. He's also made some foundational discoveries about the importance of viral capsid structural dynamics, and this has the potential to greatly inform vaccine design. He's visited Virginia Tech main campus, but this is his first time at VTCRI, so I'm simply thrilled to have him here. Please join me in welcoming Dr. Richard Kuhn.
Wow, thank you. That was a fantastic introduction, Sarah. Now I think I'm going to really have to try to live up to that introduction. So I'm very pleased to be here. I had some really great talks with colleagues yesterday and today, and I'd like to thank Dr. Friedlander for the opportunity to come and visit and tell you a little bit about the work that we've been doing. And I should say from the outset that I've collaborated with Michael Rossman, who is my next-door colleague, for 25 years. And a lot of the work has been a very tight collaboration with Michael. I should say, Sarah mentioned I was department head for way too long, 12 years, and I've just stepped down. And Michael was very unhappy with me when I became department head, and he was continuously unhappy while I was department head, with the minor exception that we were able to build a brand new building, and and most recently, I'll just say this now, our ability to rapidly solve the structure of Zika at near atomic resolution was really driven by us acquiring what's known as a direct electron detector. And Michael was so pleased to see that I personally contributed several $100,000, not me, the department. As department head, I was able to do that. And so I think he does value that time. So now I'm much more engaged in research, and I'm going to tell you about some of the work that is very recent. I will tell you about work that actually was submitted very recently on Zika, and stuff that's been going on for probably at least 16, 17 years. So flaviviruses, these are RNA, positive sense RNA viruses. 5' end has a cap, 3' end is not polyadenylated. They're very small viruses. They're in the order of 500 angstroms in size. They have a lipid bilayer, which it makes, I think, some of the structural aspects quite interesting. In the genus called flaviviruses, there are lots of members. These include viruses like dengue, which I'll spend most of the time talking about, but yellow fever, West Nile, Zika, and they cause a wide variety of clinical symptoms in humans and in animals. The family also includes hepatitis C virus, so this is a very impactful family in terms of the impacting human disease. I think we all know, I'm going to start off with telling you a little bit about Zika virus. The good thing is probably you know as much background about Zika as I do. You know, it originated from the Zika forest, and these were studies that were ongoing to look for the distribution of yellow fever. So there were sentinel monkeys that were placed, you know, at various heights in the forest, and they were able to isolate a new virus, which they named after the location that it was identified in. There was not a lot of activity in terms of human disease associated with Zika virus, and it spread. Presumably it spread to the east, entering Asia, and then really the first events in which it was recognized that it really causes some level of disease was infection in Yap Island, and then followed by now an epidemic in French Polynesia, and then the real outbreak that occurred in Brazil and has spread since, and it has spread all over the place. This was actually from a few months ago, but as you all know, it's all over the Americas right now. And so it's an interesting virus that we need to look at. You probably know as much about this as I do. The lucky thing with Zika virus, I guess, is that it's not a really, in terms of most people who get infected, it's usually not a bad virus in terms of disease. It's the typical symptoms of a virus infection for most people, and in fact, only about 20% of people show an apparent infection. And really, this 80% of inapparent infection is, I think, the thing that we should worry about more than this group right here, because these are the potential transmitters of the virus and unknown consequences from that. As you all know, Guillain-Barré syndrome has been reported for people who have been infected with Zika virus, and this is not, you know, this syndrome is not specific to Zika, although Zika certainly has a higher percentage of people showing this syndrome. The number of viruses and other pathogens can induce this syndrome. I think what really kind of raised alarm, and which is of important consequence, is the effect on the fetus if a mother is infected during pregnancy. And my own opinion is that this microcephaly is really the tip of the iceberg. I think we're going to have to wait 5 or 10 years and really see what the developmental problems of those kids who are infected in utero but don't show these kind of obvious phenotypes, you know, what's going to happen with them developmentally in the future. And so I think there's a lot of interesting biology here. Why didn't Zika cause these neurological impairments in the early period before the Brazil? Perhaps one thing is that the number of people was just not high enough to identify something like microcephaly. Some people have speculated that the virus has mutated. Certainly there are a number of different mutations have arisen. Again, I don't think there's any strong data that says that there's a specific mutation that's triggered this new disease phenotype. Whether there is, you know, people have suggested, well, this is now co-circulating with dengue, for example, and perhaps there is an antibody-dependent effect. There is no data, as far as I'm aware, that would lead to this conclusion right now, that it's an ADE-type effect with a pre-existing immunity to other flaviviruses. Now, here's the interesting thing about Zika virus. It's an arbovirus. It's transmitted by mosquitoes, and it's transmitted by primarily Aedes aegypti. This is an urban dwelling mosquito. It likes to sit in the house. It bites at all times of the day. And so this is a virus that can quickly spread locally in a family, in a community. But it's more. Zika is more than your normal flavivirus. Zika, like West Nile, can be transmitted through blood transfusions. But unlike, I think, all of the other flaviviruses, sexual transmission of the virus has been established. And this makes it not only an arbovirus transmitted disease, but a sexually transmitted disease. And so I think that adds some interesting consequences to it. Although I think most people believe that this is not a sustainable transmission model. I think the sustainability is really going to continue to be driven by mosquito transmission. Good news is that their NIH has already moved into phase 2 and probably will soon move into phase 3 clinical trials with a number. This is a DNA-based vaccine, but there are a number of vaccine candidates that the Vaccine Research Center is currently working on. And they are not alone. There are probably more than 18 fairly well-established groups that are developing different types of Zika virus vaccine. I think the earliest is probably 2018 that we might see this. By 2018, those endemic areas are those areas that have epidemic virus right now, probably will have endemic virus at that point, and that you will have a fairly high seropositive number of the population. So maybe, I'm not sure how good the vaccine will be for some of those areas. The genome of Zika, like the flavivirus, is rather simple. We make 10 proteins. Those 10 proteins are initially made off of a single polyprotein that is cleaved by a viral as well as cellular proteases. 3 of the proteins are what we're going to talk about today. These are at the amino terminal end of this polyprotein, and these are the components of the virus particle. They are the capsid protein, the membrane or pre-membrane PRM protein, and then the major protein that we have interest in, which is the envelope protein. And the envelope protein, as you will see in lots of detail, really covers the surface, the lipid bilayer of the virus particle. The envelope protein is responsible for finding the target cell. So Zika, the interesting thing about Zika biology is what is driving this unique tropism of moving across the placenta and moving into progenitor neuronal cells in the developing fetus. I think that's one of the major questions in the field right now. Besides this binding to the cellular receptor, the envelope protein is a fusion machine. So under low pH conditions, when the virus enters the cell in the endosome, the E protein oligomerizes into a trimer and is able to fuse the viral lipid membrane with the endosomal lipid membrane, allowing the contents, which is going to be the capsid protein and the genome RNA, to be released into the cytoplasm. And a positive strand RNA virus, for those of you who are not so familiar, if you just take that RNA is just like messenger RNA and will translate its proteins. So for this type of virus, all you really need to do is introduce this RNA into a cell and you initiate infection. So it's really quite easy. So let's just talk a little bit about what we did for Zika. We really dove in very deep. We were working, actually at that time, we were working on a number of flaviviruses and trying to compare structures. And so it was relatively easy to move into Zika. But early, when we started this work, there wasn't a lot published information on large-scale preparation of the virus. And so we really had to work out a lot of growth conditions and purifying sufficient amounts for our cryo-electron microscopy. And as I kind of hinted at already, the setup we have with our electron microscope allowed us with Zika to take the EM density that we were able to derive and then build directly the atomic structure of the virus. And so that's relatively different than what we've done with all of the other flaviviruses, in which we combine crystallography of the component proteins and put them into a structure of our EM density. So this, we built it de novo, if you will. So we looked in a variety of different cells. Eventually, we used a cell that we were working with our collaborator, Ted Pearson from NIAID. And Ted had taken Vero cells and introduced furin. So these cells overexpress furin, and I'm going to tell you why that was important probably definitely later in the talk. But those gave us the best preparation of virus particles, and we used our Titan Krios together with, as I said, a direct electron detector. Now, the beauty of this system is that it is so sensitive that you actually have vibration caused by the electrons passing through your sample. And so instead of taking a single image and using that, you actually take a whole series of images, you make a movie, and then you computationally correct this vibration that you see using multiple images of the virus particles. So our ability to go to atomic resolution, which is really, for those of you who are not in the structure field, it is a revolution in structural biology to be able to use electron microscopy. And Michael Rossman worked with Max Perutz. I mean, he is well known as a crystallographer. He will tell you, for the most part, that crystallography is a thing of the past, and that electron microscopy is the way to go for most things, not everything. But Michael essentially would always do EM if he can. This is an image of Zika. This is mature Zika virus from our cryo-EM. You can see that the vast majority of these particles, I think you can see the vast majority of these particles are fairly homogeneous and smooth in appearance, and that is consistent with what we would normally refer to as mature flaviviruses. And so from that, we were able to solve the structure of the virus. This is looking at the surface of the virus. We've colored it based on its distance from the center. So what you see on the most peripheral part of this is in red, and followed by yellow, and there's blue, and there's a whole gradation of colors. A couple of observations. One is that you don't see the lipid bilayer in this structure. Okay? It so happens that in the flaviviruses, unlike HIV or flu, in which their glycoproteins are perpendicular to the lipid bilayer. In the flaviviruses, these proteins lie flat, at least in this form of the virus. I'm going to talk about different forms of the virus in detail later. But the lipid bilayer is not accessible, which is interesting because dengue virus and Zika virus, they have been suggested in the case of Zika, to use certain types of receptors that bind phosphatidylserine. So where is the phosphatidylserine in terms of binding to a receptor? I'll get to that a little bit later. What you see here is part of the E protein and part of a glycan that sits on the very periphery of the E protein. So that's what this little small projection off the top of the virus particle. If we look in cross-section, you can see the layered nature. So the E protein and, to a much lesser extent, the M protein, because it's very small, make up this very thin and flat surface on the virus particle. They are both transmembrane proteins that are anchored in the lipid bilayer. And the bilayer here, in green and in blue here, or aqua, yeah, I guess that's close enough. Cyan. I am not very good with colors. This is the outer and the inner leaflet of the bilayer. And I think you can see in this view, these things that cross it, these are the E and the M proteins. Another thing to observe is that these don't have cytoplasmic domains. They basically hit the inner leaflet and then they turn back on one another. And so they're true anchors. And they're really anchors because they're fixed in the virus particle. Otherwise, we wouldn't get them seen in this reconstruction. Okay. This is the inner core, which is the capsid protein and the RNA. And right now, I would say that this is not very well organized in terms of our ability to solve the structure. Just a caveat, we just have a paper accepted in Nature Structure Molecular Biology yesterday in which we have the immature structure of Zika virus. And one of the amazing things that we see in that structure is that we now see an partially ordered core. And that's the first time we've seen that for any of the flaviviruses that we've been looking at. So this is the C-alpha backbone of the E and M proteins, and you can see that this is an icosahedrally organized virus particle. And I'm going to apologize. I'm going to tell you in a few minutes about icosahedral structure, and you're probably not going to be very happy with that. But I think it has interesting consequences for the biology of the virus. But you can see this organized structure here. You can see that there's this aggregation, if you will, or oligomerization of this blue domain. This is an Ig domain. I didn't go into the structure of the E protein very much. but this is an Ig domain. It actually sticks up a little bit along with the glycan. The glycan is here, and this is some of the loops of the E protein from this Ig-like domain. Ig-like domain has been implicated in receptor binding for a number of the flaviviruses. But you can see that this actually turns out to be what we would refer to as a five-fold axis of symmetry. Right here, there's a threefold. You can see 1, 2, 3. These are related by a rotation of 120 degrees. And then right here, we have two molecules of the E protein, an E protein dimer, and these sit on a twofold axis of symmetry. So if we rotate this particle by 180 degrees, it's identical. Okay, that allows us really to drive resolution, and it has consequences, important consequences for the biology of the virus that I'll talk about in a few minutes. This is now a side view of the envelope dimer and the M dimer from our structure. You can see that the glycan kind of sticks out here, and this loop right here that's coming from this domain 3, this Ig-like domain. This region is on the outside covering the bilayer. These are the transmembrane domains, so this is in the bilayer of the virus itself. So you can see that these viruses expend quite a bit of protein currency in terms of embedding themselves in the membrane. And these, as I said a few seconds ago, these are fixed. They don't float around as you might imagine transmembrane domains do. They're fixed, even though they're not anchored on or held on the other side of the membrane. They're fixed in place. Okay? So this is a larger view of the structure. So M, PRM, is an important protein for maturation of the virus. The M protein, we don't know really what function it plays in the mature virus. We know the function of the precursor protein, but not of M. M doesn't even extend out. In fact, you don't really develop many antibodies against the M protein in normal infections. It's really hidden. The majority of the protein is actually in the anchor region here. Okay. Now, Zika, if you look at percent identity of these proteins, PRM and the E protein, they're not that high compared with the other flaviviruses. And what I'd like to do is just compare Zika with Dengue 2, which is the virus that we have the most structural information from, and it actually is interesting from a vaccine standpoint. 54% of the E protein at the amino acid identity level. And if we look, we take the C alpha backbone from our dengue structure and superimpose it on the C alpha backbone of Zika virus, you can see that even though they're only about 54% identical, they superimpose exceedingly well, okay? Very well. Even in these transmembrane domains, they pretty much sit in exactly the same position. There are a few exceptions. There's some variability over on this end and right here, and there's quite a bit of variability right here. And that variability is blown up here on the right. That variability is right at that glycan. Now, the interesting thing about this region, this is now a loop region, and the loop is clearly very different between all of the flaviviruses. I'm showing dengue against Zika right here. I'm going to just go back a second. Oh, I guess I have to go back 2 seconds. It's actually a little bit difficult to see, but I told you this is actually a dimer of E protein, okay? And so this loop that I just was talking about and the glycan sit here, okay? Now, this loop actually sits on top of this green loop. The green loop is a fusion loop. That's the residues that are involved in facilitating fusion of viral and cellular membrane. This loop is actually coming from the adjacent monomer. And what we think, and this is based on a lot of data from Ted Pearson and Mike Diamond, in terms of antibody accessibility in this region, we think that this loop is really a flap that flaps up and down and allows access of the fusion peptide to the environment, okay? And this is a region that there's quite a number of antibodies that are directed against this region. I should also point out, in the situation with West Nile virus, the presence of this glycan at residue 154 has a significant effect on whether the virus is neurotropic. Okay? So from what I've told you then, we suggested that this glycan and this loop play an important role in entry of the virus. It may also play an important role, but we're not as certain about this, in attachment or in tropism of the virus. Okay? And Ted's lab now has evidence that it's clearly involved in the entry process. And this loop region, you can see, is very different among the different flaviviruses, okay? And you can see that there are differences even in Zika, whether there is a glycan at position 154. But this right here is that loop exactly, and we think that this is one of the control mechanisms for viral entry. I'll just point out, because of the fact that you have 3 identical proteins in what we call the asymmetric unit on this icosahedron, and those are repeated 60 times. But those 3 positions, identical, chemically identical proteins, right, the E protein or the M protein, they sit in different environments relative to their neighbors. And so they have slightly different positions based on whether you're at a 5-fold axis, close to the 5-fold axis, or the 3-fold axis, or the 2-fold axis. And you can see that there is differences in this loop. And I'm going to come back to that in a second. I'll just let you run through a little movie showing the virus surface. This is what we call the raft. It's a set of 3 E dimers that line up together. Now this is the single E protein dimer, showing it's shaded, rotating around. I'm not sure why we go upside down with it. Okay, so now this is looking down on it, and this is looking from the side. You see it's upside down relative to this is where the membrane is going to be right here. You can see that this region right here is clearly sticking out and would be a great attractant, perhaps, to bind to a cell. And then we'll just put this thing back into the virus particle. You can see it. You can see that dimer. This is looking down the 5-fold, and you can see rotation of the virus. Oops. Okay. So let me just change for a second. We have submitted this paper. This is actually. Let me back up. This is work that was done by Jim Crow at Vanderbilt and Michael Diamond at WashU. They were able to isolate a human Zika antibody, and they were able to produce it as a monoclonal antibody. It is a highly potent neutralizer of activity in cell culture, and in a paper that was released last month in Nature, they were able to show that this antibody is protective in mice against Zika virus infection. And so what we have been doing is looking at the structure of how this antibody, which is called Zika V117, binds. And the data suggests that it affects multiple stages of the entry process of the virus. This is now a cryo-EM reconstruction of Zika virus complex with 117. You are the first audience that I'm showing this to. This is this asymmetric unit, this repeating block that icosahedron has. And the antibody binds to the domain 2 of the E protein. There are 3 E proteins that are located within this asymmetric triangle. But when we look at antibody, or Fab binding in this case, when we look at Fab binding to the E proteins, we actually find that only 1 of the 3 potential sites is occupied by a Fab. And so you have to ask the question, you have to ask a number of questions. 1, why? And 2, then how does Fab neutralize? Fab, in fact, does neutralize the virus. So you don't need intact antibody to neutralize Zika virus with this. So what I'll tell you is that the majority of binding 2/3 of antibody or Fab binding occurs at this site right here, which is we call a 3F because it's close to a threefold axis. 1/3 is found close to this twofold axis. And there's this very, very little binding that occurs at this site, which was close to the fivefold axis of binding. Now here's the part that you're probably not going to like so much. Oh no, this is not. You'll love this. This is just a little. This is just a little. Let me show you. We have these 3 E proteins that are in different chemical environments. We have them colored in red and gold and blue. Okay, you can see this is a nice fivefold. You can see that this is also a threefold, and this, I will tell you, if you can't already see it, is a twofold, okay? So those are the axis of symmetry for the virus. And just to kind of show you as it moves around a little bit, okay? Fine. Okay, here's what you wouldn't like that much. But this is really, from our standpoint, this is very interesting. And I'm not going to spend too much time because you really should be a little nuanced into virus structure. But this is the main site in which we see Fab binding right here. It's sitting on this site right here. There's another site here. There's another site over here. Now, there's no binding to this site over here. There's a little binding to this site, and most of the binding is occurring on this site right here, okay? And the reason is that the epitope is fully exposed at this site, mostly exposed at this site, and not really exposed at this site. In fact, this site is blocked by the neighbors. You could imagine that this site is in a canyon and that antibodies cannot penetrate into the canyon because they're blocked. There's a big ridge that's right over here. This one here is pretty much open, and so antibodies will tend to bind to this one first. If they bind here, they can't bind here. But the reciprocal is also true. This is a lower affinity binding site, but if you do bind a Fab here, then you cannot bind it here, okay? And now the question is, how many antibodies do you need to bind to neutralize virus infection, and how does it work? It works simply by when the antibody binds, or the Fab binds, it actually binds these dimers together. They're no longer able to rearrange, which is required for entry into the cell and low pH mediated fusion. Because these guys, these dimers, are going to rearrange into trimers. That's what's required for low pH fusion. And the antibody binding to it locks up the whole virus particle. And we have predicted now that the number of binding sites that you need to neutralize this virus is going to be very low. So you have a high affinity binding site plus a low number of antibodies required for neutralization, and that's why we think that this is such a potent antibody. This is a roadmap showing kind of the surface features of the footprint of where the antibody is binding on the surface of the virus. And this is a very high hill that prevents binding on this site right here. This site is not as clearly exposed as this site. So that's antibody. I'm going to switch and talk in the remaining period about dengue virus. Dengue is very closely related to Zika virus, and unfortunately, in many of the areas where you have Zika, high levels of Zika, you also have lots of dengue virus. And there's also the alpha virus, chikungunya virus, which we won't even talk about today. So as I mentioned at the beginning, this is a virus cloud showing the differential diseases that are caused by the various flaviviruses. And although Zika has emerged with a real pounce, dengue is still the most important of the flaviviruses. They cause a variety of diseases. We've heard about Zika virus, but of course you're also familiar with West Nile virus, which is a neurotropic virus, yellow fever, a hepatotropic virus, and then we have dengue virus, which causes some very interesting diseases. My lab has been interested in how does virus structure, the heterogeneity that I'm going to talk about, and the plasticity or dynamics of the virus particle, how does that influence tropism and disease? And I will hopefully quickly move through this. So, the drawer is always Zika virus for the last few months, but dengue virus is still the most significant arbovirus problem in the world. There are 4 dengue serotypes. There has been a licensed vaccine last December, so 1 year ago. It's licensed in 5 countries at this point. It's a vaccine from Sanofi Pasteur. I will say that we have worked with Sanofi Pasteur, but it's not the best vaccine because of the 4 serotypes. Serotype 2 is a very ineffective vaccine. That one does not provide nearly complete protection. And there is the question of whether the vaccine might cause more problems than prevent. Okay. So about 400 million infections worldwide per year. And this is just an explanation of the 2 types of disease. You have kind of the normal dengue fever. It's not a great thing. If you get it, you probably feel like you would prefer actually to die rather than go through it. That's what people will say, although again, it's probably only about 80%. It's only about 20% of people get an apparent infection, but that's a rather nasty infection. But it could be worse. You could come down with dengue hemorrhagic fever, dengue shock syndrome, in which you have significant capillary leakage, and if it's not managed correctly, there is a chance of dying from this. And most people are of the opinion that this is primarily caused by sequential infection with differential serotypes. So if you are infected with dengue, you'll get nice protection, lifelong immunity against that serotype, and so if you are infected again, there's no problem. But on the other hand, depending upon the sequence, if you are infected with one of the other serotypes, there's a potential for a secondary heterotypic infection that is severe. And antibody-dependent enhancement is believed to be the mechanism by which this occurs. You have antibodies from the primary infection that cannot neutralize the virus but bind to the virus particle. Those are then recognized by Fc-bearing macrophages, internalized, and virus is released, causing an infection and eliciting essentially a cytokine storm. And so that's the result of that is the hemorrhagic disease. Very similar to what I've just talked about in terms of Zika virus. For dengue virus, we solved the structure of dengue virus 16 years ago or so, maybe 15 years ago. And at that time, we called dengue virus a golf ball, very nice structure. We thought it was great. We did a whole bunch of studies to look at the maturation process of the virus. And we showed, and I'll briefly go through this, that the virus undergoes very significant transitional and translational conformational changes during the maturation process. And so we were quite happy with that. But we've now realized that it's much more complicated than we first thought. It turns out that this process of virus maturation, for dengue in particular, is very inefficient. And so we talk about viruses that don't grow up. And that has, we think, significant consequences for disease. We've also been able to show, and this is work that we've done with our collaborators, Mike Diamond and Ted Pearson, that using antibodies primarily, that there is a significant amount of dynamics that these virus particles are in constantly undergoing. And that also has consequences for disease. And in at least one case, we found that the virus has a different structure depending upon the temperature. So another consequences. This is, of course, very closely related to this concept of dynamics. So the virus is first produced by budding into the ER lumen in an immature form. This structure of the virus we have solved, and as I mentioned, we just published this, we just got the paper accepted for Zika virus. And so it is formed in this immature state in which you have PRM bound to E as a heterodimer, and 3 of those form a projection on the surface of the virus particle. This particle then moves through the secretory pathway, and when it enters the trans-Golgi, that's in a slightly acidic environment, and that acid environment causes a rearrangement of the viral proteins. And so now these guys all lie flat. And when that rearrangement occurs, the furin cleavage site, which was inaccessible in this form, now becomes accessible, and you can carry out cleavage using host protease furin. The PR component is not released until you hit a neutral pH, presumably when the virus is released from the cell. Okay? So this is the process of maturation, shown slightly differently here. Here's the structure. Here's the C alpha backbone that you can see. It's standing up. It's laying down. It's being cleaved. This component PR is being released when the virus comes off the cell. This is a non-infectious form of the virus. This is an infectious form of the virus. Okay? Now, it turns out that in dengue, this is very inefficient. So if you look at virus preparations from dengue virus, you find that there is a whole series of different types of viruses that are found in the supernatant. And when we originally did the structure, being nice, good structural biologists, we threw away everything we didn't like. We looked for things that looked the same, okay? And smooth things obviously look smooth. And that's what we did, and that's what we said. But it turns out this is the usual. Now, you might ask, cell culture, it's an artifact. But in fact, and I'll just mention this in a few seconds, people who are infected with dengue, and in fact, people who have dengue hemorrhagic fever, have very high levels of antibodies against PRM. And so, although we've really been trying to look at virus released from infected patients, it's usually, the best, the people who are producing the most virus are the hemorrhagic patients. It's probably not a great idea to draw a lot of blood out of them. It's very, very likely that there is this mixture in infected patients because of the antibodies. Not only that, but these antibodies have interesting activities. Some of them have neutralizing activities, okay? So it's very clear to us, at least, that this observation that we see in cell culture is probably true in infected individuals also. Now, with dengue 2, we have a smooth virus particle if we keep it at room temperature. If we elevate the temperature above 33 degrees, we actually have an expanded virus particle. And this is a stylized view of what we think is happening. So you shouldn't think of it as a golf ball. That was completely wrong, although it was easy to explain to people. We should think of the virus particle as this thing that is living and breathing. Not living, but breathing, okay? And this certainly complicates our understanding of the structure. We've been using both cryo-electron microscopy as well as we've developed a method for quantitatively measuring the amount of cleaved PRM on virus particles. And we've been looking at a variety of different flaviviruses. It turns out that dengue 2, which is the one that we've studied from the get-go, this is the one that actually turns out to be the most heterogeneous of the flaviviruses. But I will tell you that even Zika virus, there's significant heterogeneity. So even though those virus particles look smooth, we now know that they do have significant amounts of uncleaved PRM in the virus particle. So there's just a comparison between dengue 2 and dengue 4. I'll just say, not revealing any trade secrets or anything like that, but dengue 2 is the component of the Sanofi vaccine that is not very good, okay? So this is just an analysis of dengue 2 versus dengue 4. Dengue 4 particles are much more homogeneous. You can look at the distribution of these. This is the cryo-EM analysis. We've counted particles and compared them based on their morphologies. We've also done mass spectrometry analysis on them. And if you look at dengue 4, you see that this PRM cleavage by furin is greater than 80% in this particular strain, and most strains that we've looked at. Whereas if you look in dengue 2, and this is our laboratory strain that we normally use, less than 50% of PRM actually gets cleaved. Now, you shouldn't think of it as simply 50%. You should think of it on the virus particle as, you can have either 100% cleaved or no percent cleaved, and everything in between. And now you can distribute that in the 180 positions on the virus particle. You're introducing tremendous heterogeneity in the virus particle. Now, that could be a challenge if you want to develop a good vaccine, for example. Now, I'm going to end with just a few words about furin. Furin is important for a number of pathogens. HIV, it's required for HIV. It's required for a whole bunch of them. It carries out this maturation cleavage. Furin binds substrates, and of course, furin cleaves mostly cellular substrates, okay? So viruses are taking advantage of this protease that is present in the secretory pathway. Very basic residues are found in the cleavage site of furin. In particular, this P3 position also prefers a basic residue, but that's one that differs in the flaviviruses. So you can see at the P1 and P2 positions, there are positive residues for all of the flaviviruses. But at the P3 position, a lot of the flaviviruses, they don't have positive basic residues, but they're okay. I mean, they're not bad. I will tell you that most cellular substrates have basic residues at this position, okay? So flaviviruses in general are not optimal at the P3 position. The dengue viruses, they are even worse. Not only do they not have a basic or a neutral residue, they have an acidic residue at this position, okay? And so we think that this is a major determinant. There are other determinants in the cleavage site. We might you might say, well, why is dengue 4 better than dengue 2 since they both have acidic residues? Dengue 4 has this arginine sitting here, whereas the other dengues have histidine, and we think that makes a difference too. And so dengue 2 has a glutamic acid here as opposed to the aspartic acids that are found in 1 and 3. So those might be differentiators that we are looking in using genetics at this point. So I'm going to just leave you with this question. Just like HIV or Hep C, the flaviviruses are RNA viruses that mutate very rapidly, okay? So why do you maintain an inefficient cleavage site when you could simply, optimize that cleavage site and get 100% cleavage or close to 100% cleavage? We would argue that there is a biological reason for the virus to do that. Attempts to mutate that, the virus does have differential defects when you mutate that site. The people who've done those studies have not identified what exactly that defect is, but we're looking at that right now. And so let me just close by saying that this question of PRM content influences the structure and dynamics of the virus particle. Again, the surface of the virus can vary so dramatically depending upon the PRM content. And that's going to influence the structure. It's also going to influence the dynamics. And I didn't talk very much about the dynamics, but we think that that's a major factor. I'll just step back 1 second, and I will tell you that our glimpse into dynamics occurred when we tried to do a structure of a neutralizing antibody against dengue virus. We took that antibody and we added it to the virus at room temperature, and it would not bind to the virus. And we were somewhat dumbstruck by how could a neutralizing antibody not bind virus. And we then did the structure, actually this was Dave Fremont at WashU, the structure of the antibody as well as the E protein, and we found that that epitope actually wasn't accessible in the mature virus as we had it. And so what we did was, of course, neutralization is not done at room temperature. Neutralization is done at 37 degrees. We then incubated virus and antibody at 37 degrees, and then we got antibody binding. And the reason is that the virus expands, and the dynamics of that particle increases as you increase the thermal energy, and the antibody binds and captures the virus in that state and then locks it up into that state. So that's how we originally found this concept of dynamics. So I think it's very important. And that influences, oops, I guess that was the signal to stop. Let me see. I probably hit the black screen. I got it. I hit the black screen. This structure and dynamics affects the infectivity of the virus particle. It certainly plays a role in entry. If you have a lot more virus surface, diverse surface, you increase the possibility that you now can go after different types of receptors, right? This clearly is greatly affecting the immune response, and certainly this has to be called into question when you're thinking about vaccines. And the major question that we have is, how does it influence disease and pathogenesis of the virus? And so that's what I will leave you with today. So I would like to acknowledge, first of all, my long-term collaborator, Michael Rossman. We've had a great collaboration. Just about 90% of the work that I showed you today was done by a very bright graduate student, Devika Sarohi. Devika got her PhD last week, which was very nice. But the bulk of the work, both the Zika and the dengue virus work that I talked about, was done by her. In the Zika structure in particular, and a lot of the other structure stuff, she worked with Shengguo Chen, who was a joint postdoc between Michael and myself. Lei Sun in Michael's lab actually was the one who built the atomic model. Thomas was really involved in getting the microscope and maintaining the microscope so that we could do all of these experiments. Andy Miller has been growing large amounts and preparing all kinds of different biological samples for our structure analysis. And Saif Hassan is the postdoc in Michael's lab that just did this work on binding of antibody 117, which again is a collaboration with our lab and Mike Diamond's lab at WashU and Jim Crow's lab at Vanderbilt. And my long-term collaborator, Ted Pearson from NIH. Ted is not smiling as much because he now has to answer Tony Fauci on a daily basis because Ted's doing all of the analysis of the stuff coming back from the Vaccine Research Center and samples all around the place. But I will make Ted smile because I'm meeting him later today in Washington, and we're going to go back to Purdue and spend a few days doing some science and other things. So with that, I thank you for your attention.